Good morning. A very warm welcome to all our distinguished guests, speakers, and delegates who have joined us from different parts of the world. On behalf of KMCT Group of Institutions, it's my pleasure to welcome you to the international webinar on One Health: Threats, Trends, and Solutions. Kindly ensure your microphones remain muted, and we request all participants to use the chat box for queries during the sessions. Once again, a warm welcome to everyone. Before we move on, here is a brief introduction to KMCT Group of Institutions. Founded with a mission to educate and empower the youth, KMCT stands today as one of Kerala's foremost educational community. Under the inspiring leadership of Dr. K.M. Nawaz, KMCT has been a beacon of excellence across diverse disciplines: medicine, engineering, pharmacy, nursing, allied health science, management, law, and more. Today, KMCT has more than 25 institutions spread across 7 campuses in northern Kerala. KMCT continues to build national and international collaborations, strengthening our mission of empowering the society through knowledge. As a part of this commitment, we are organizing this One Health webinar, which recognizes the deep interconnection between human health, animal health, and environmental health. Through this initiative, KMCT aims to promote interdisciplinary collaboration and research. By bringing experts and institutions together to one platform, we hope to contribute meaningfully to the global One Health movement. To formally welcome you all and to give an introduction to today's webinar, I invite Dr. Santosh VC, Chief of International Affairs, for the welcome address. Dr. Santosh, please.
Respected dignitaries, invited speakers, faculty members, students, and participants joining us from India and across the world. It's my honor to warmly welcome you all to today's online webinar on the One Health concept, organized by KMCT Group of Institutions. This webinar reflects our collective commitment to understanding the vital and inseparable link between human health, animal health, and environmental health, the very foundation of the One Health approach. As the world faces emerging zoonotic diseases, antimicrobial resistance, climate change challenges, and ecological imbalances, the One Health framework is no longer an option, but a necessity. Today's session aims to deepen our awareness and strengthen interdisciplinary collaboration toward a healthier and more resilient future. We are privileged to have an esteemed panel of 5 distinguished resource persons, each contributing significantly to the global One Health ecosystem. Dr. Arun T. Ramesh, scientist at ICMR National Institute of One Health. Dr. Muhammad Elayadat Meethal, Research Fellow at University of Melbourne, Australia. Dr. Deep Jyoti Bhuyan, Senior Research Fellow at Western Sydney University, Australia. Dr. Minu Divagaran, Director of Research and Consultancy at Providence Women's College. Dr. Anish T.S., Professor of Community Medicine and the Nodal Officer of the Kerala One Health Center for Nipah Research and Resilience. We are truly honored to have each of you with us today. I also extend my heartfelt appreciation to the KMCT leadership, organizing team, faculty members, and participants whose efforts and enthusiasm have made this webinar possible. I hope today's session will inspire thought-provoking discussions, create awareness, and encourage collective action under the One Health framework. Let us make the most of this opportunity to learn, share, and strengthen our understanding of holistic health. Once again, a very warm welcome to each one of you. Thank you, and I wish the session all success.
Thank you, Dr. Santosh. Now, I have the pleasure of inviting the guiding force behind the KMCT Group of Institutions, Dr. K.M. Nawaz, our chairman, for the presidential address. Sir, please.
Good morning, distinguished delegates, esteemed speakers, respected colleagues, and dear participants. On behalf of KMCT Group of Institutions, it gives me immense pleasure to welcome each one of you to this international webinar on One Health. It is an honor to address this the gathering of experts, scholars, practitioners, and visionary leaders from national and international organizations and universities. Today's event is more than an academic exercise. It is a collective commitment to a safer, healthier, more sustainable world. The concept of One Health reminds us that the health of humans, animals, and environment are inseparably linked. As we confront global challenges such as emerging infectious diseases, antimicrobial resistance, climatic changes, the need for the resilient health systems, the One Health approach has never been more relevant. At KMCT Group of Institutions, we are deeply committed to advancing interdisciplinary education, research, and collaboration. With our vast number of colleges in the field of healthcare, working together with other umbrella institutions, we are uniquely positioned to translate One Health principles into impactful action. This webinar marks yet another step in strengthening our global partnerships and expanding our academic vision. I sincerely thank each of our distinguished resource persons for accepting our invitation and contributing their expertise. I also appreciate the dedication of our organizing committee, faculty members, staff, and students who have worked tirelessly to make this event possible. I hope today's discussions ignite new ideas, foster meaningful collaborations, and motivate us to build a healthier future through integrated multidisciplinary approaches. With great enthusiasm and optimism, I officially declare this international webinar as open. Thank you very much. I wish you a productive and insightful session. Thank you very much once again.
Thank you, sir. May I now invite Dr. Vijesh Venugopal, Principal, KMCT Medical College and Directorate of Research, KMCT Group of Institutions, to speak a few lines.
Respected Chairman, esteemed organizing committee members, distinguished resource persons, dear participants, namaskaram and a warm welcome to this virtual gathering. It's my profound honor and privilege to extend heartfelt felicitations and congratulations on behalf of our entire research community, the organizing team of this webinar led by Dr. Santosh VC and Dr. Jayakrishnan T for planning this timely and apt international webinar on One Health, threats, trends, and solutions. In an era where boundaries between human, animal, and environmental health are being increasingly blurred, the One Health approach stands as a beacon of integrated wisdom. This webinar beautifully encapsulates the essence of unifying efforts at local, regional, national, and global levels to recognize the interconnections that shape our collective well-being and for the bright future of our ecosystem. By bringing together experts to explore the interdisciplinary dimensions, that is from antimicrobial resistance and zoonotic diseases to climate change and biodiversity, we are not just discussing challenges, we are forging strong collaborative solutions that can contribute to a healthier and a more sustainable world. I am extremely delighted to the stellar lineup of speakers who have come here today to share their valuable time and their expertise with us. And this will undoubtedly enrich our understanding and inspire actionable research on the theme of One Health. To all participants who are joining us from across the globe, your presence here underscores the universal relevance of One Health. So make the most of this platform to learn, interact, collaborate, and contribute to the concept of One Health. On behalf of the Directorate of Research, I wish this webinar resounding success. May it pave the way for stronger partnerships and a more resilient future for our planet. Thank you. Jai Hind.
Thank you, Vijesh sir. Before we move on to the scientific session of the day, let me introduce Dr. Jayakrishnan T, who will be moderating the entire session today. Dr. Jayakrishnan is a professor and head of the Department of Community Medicine at KMCT Medical College. Dr. Jayakrishnan brings over 30 years of experience from various government medical colleges and currently serves as the Secretary of the Institutional Ethics Committee at KMCT. He has held prestigious positions such as Head of the UNESCO Bioethics Chair, Coordinator of the Regional PEID Cell, and Convener of the State Technical Advisory Group in the health sector for the 10th and 11th five-year plans. He also serves as the Vice President of the Indian Public Health Association Kerala chapter. I welcome you, sir, to the webinar. We will now move on to the scientific sessions of the day. To begin with, we have with us Dr. Arun T. Ramesh, Scientist C at the ICMR National Institute for One Health, Nagpur. Dr. Arun is a veterinary professional and has previously served as Scientist B at the ICMR National Institute of Virology, Pune. He is the co-investigator in several One Health projects and has also served as a former member of the Veterinary Council of India and the Board of Management, Kerala Veterinary and Animal Sciences University. Today, Dr. Arun will be speaking on a highly relevant and a timely public health theme, One Health, Threats and Preparedness in India. With high respect, I invite Dr. Jayakrishnan to kindly present our speaker and his topic. Thank you, sir. Over to Dr. Jayakrishnan.
Good morning, everybody. As mentioned by previous speakers, One Health meaning interconnected health. That means it's called for work together across sectors. Taking the above facts, our speakers include scientists, doctors, veterinary science, researchers from pharmaceuticals, plant science, biologists, etc., all around the world. We hope the discussion today will give us clear, measurable solution in future. And coming to the point, geographically, India lies in tropical region with a lot of climatic threats. Being the most populous countries in the world with 1.46 billion people with a lot of biodiversity and humans are living mixing with animal population, India are vulnerable to many infectious diseases. Perceiving this, Government of India in 2019 started One Health Center at Nagpur as a satellite to NIV Pune under ICMR. And our speaker, today's speaker, Dr. Arun is involved in the sentinel surveillance activities of this center under One Health in whole of India. And he will speak about what are perceived threats for India and experiencing trends, what are the actions to be done in India. I invite Dr. Arun to speak this session.
Thank you, sir.
Arun, you can change to slide mode,
Yeah, sure, sir.
presentation mode.
Sure. Is it visible, sir?
Yes, visible. Yes, visible.
Okay.
You can start.
Yeah. Good morning, everyone. First of all, I would like to thank Dr. Jayakrishan sir and the organizing committee of this event for giving me this opportunity to talk about One Health challenges and preparedness in India. So it was in 2018, after the first Nipah virus outbreak, a group of public health experts in Kerala, including Dr. B. Iqbal sir, Dr. K.P. Aravindan sir, Dr. T.S. Anish sir, who is also speaker today, and Dr. Jayakrishnan sir, and I was lucky to be part of that team. We started a campaign for One Health. And in 2020, under Kerala Shastra Sahitya Parishad, we organized a 1 year campaign for One Health in Kerala, which culminated in around 4,000 classes in different colleges, schools, and public health libraries and different platforms in India, in Kerala. So, yesterday, while I was attending the Virocon conference, the PSA fellow Dr. Sindhura Ganapati was he was praising the advancement that Kerala has made in One Health landscape. So I believe that the public health campaigns or the awareness that was created through different campaigns in last few years has helped Kerala in advancing in One Health. So I think the like creating awareness and talking about One Health is very important to advance further in One Health preparedness for India. So coming to the topic, so as many speakers have already mentioned, One Health is all about recognizing the interconnectedness between human health, animal health, and environmental health, and it's about working at local level to the global level, and it's all about having a transdisciplinary approach. It's not about people working in different silos and communicating with each other. It's about working together. And under the One Health umbrella, most of the public health concerns, including antimicrobial resistance, environmental health, food safety, vector-borne diseases, and zoonotic diseases, even non-communicable diseases, comes under the broad umbrella of One Health. So when we look into the history of One Health concept, it has a legacy of that goes back to Dr. Rudolf Virchow, who was the father of modern pathology and the founder of social medicine, who introduced the concept of one medicine. And Dr. William Osler, Dr. Karlin Schwabe, these are the individuals who have contributed immensely to the concept of One Health. So, even though that concept of One Health was there centuries back, it was during the SARS outbreak that happened in 2002 to 2004. Uh, the global health community recognized the importance of One Health, and in the coming years, we witnessed continuing threats of avian influenza. And in 2006, a One Health initiative task force was created by American Medical Association and American Veterinary Medical Association, and there was a formation of One Health Commission in 2007. Later, WHO, World Organization for Animal Health, and FAO joined hands for a One Health tripartite assembly. But as all of us know, it was COVID-19 pandemic that created a new attention to the concept of One Health, because there is a lack of clarity on the origin of the pandemic, but we can definitely say that it came from a wild animal. It's not only about the origin of the pandemic, but during the journey of COVID-19 pandemic, we have witnessed that the SARS-CoV-2 virus has jumped into multiple animal species, and according to the current update, there were around 24 different animal species has been infected with SARS-CoV-2 virus. And there were huge outbreaks of SARS-CoV-2 in wildlife in different countries. And there were reports of the virus jumping into wild animal, and again a reverse zoonosis happened to humans. So it's not about COVID-19 pandemic. When we look into the history of pandemic, we can see that most of the pandemics were created by zoonotic threats, including the plague, avian influenza, HIV. And so the spillovers, jumping of pathogens from humans to animals and from animals to humans, that happened throughout the history. And you can say that like human history is about the history of human struggle against the ongoing threats from zoonotic diseases. So when we talk about the challenges, there are multiple factors, multiple drivers that contribute to emerging zoonotic threats, including the high population growth. And there is a huge increase in the food animal production all over the world. And there is a huge changes in the biodiversity. As you can see in the picture, like before the existence of humans, and after the expansion of human population, there's a huge change in the uh, biodiversity landscape of our planet. The human biomass, it has grown 8.5 times, and the domestic animal biomass, if you look at it, it has grown 14 times. So there is a huge change that has happened in the planet, and there is increased global travel that has facilitated the transmission of pathogens to crossing country borders. And still, we have a lot of wildlife markets, illegal wildlife trade that's happening. And when we look at the specific challenges in India, we have a high population density, and we are witnessing a rapid urban expansion. There is a close proximity of humans, livestock, and wildlife in India. There are a lot of different interfaces that exist in our country. There's huge biodiversity that's there in our forests, and there is an expanding wildlife interfaces and ecological disruption that's happening. And we have a very huge livestock population with varied biosecurity measures, and the uh, huge close proximity that's happening in between the livestock and humans. And there are regional disparities in our laboratory and genome capacity. There are weak infection prevention and control strategies in different settings, and there is a high burden of endemic zoonotic diseases in India, and we are facing a challenge of antimicrobial resistance that is intertwined with zoonotic cycles, and we are facing a constant threat of climate change associated issues, and there is a limited structured wildlife and vector surveillance in India, and there is a insufficient risk communication and misinformation challenges that we are facing, and there are a lot of biosecurity gaps in live animal markets and animal movement. So when we talk about One Health challenges, the primary challenge we are facing is zoonotic diseases. And as all of you know, 60% of the non-infectious diseases are of zoonotic origin, and 75% of all emerging infectious diseases, and 70% of those infections come from the wild animals. So all the priority pathogens identified by World Health Organization like CCHF, Marburg, Nipah, Zika, all are of a zoonotic origin. And even when we see the list of uh, most common communicable diseases in Kerala, we can identify that three-fourths of those are zoonotic agents. So when we talk about zoonotic challenges, it's like much more complex than what we think. When we talk about Kaiserland forest disease, we can think about monkeys, humans, and the ectoparasite. But the actual situation, there are multiple animal species that are involved in the transmission cycle of Kaisano forest disease and the ecological disruptions and climate change that has to play a big role in many of the zoonotic diseases. And we are facing the silent pandemic of antimicrobial resistance, which is one of the greatest challenge public health threats of our era. And slowly we are going back to a pre-antibiotic era as most of the common pathogens are gaining resistance against the commonly used antibiotics. And it's predicted that we may be facing 10 million deaths each year by 2050 because of antimicrobial resistance. So here, the animal husbandry sector and the environmental interfaces has to play a big role in transmission of antimicrobial resistance. So definitely the answer lies in a One Health approach. And there were like some isolated attempts from different ministries and departments in India. ICMR was having some One Health projects. Department of Biotechnology was having a One Health consortia all over India. Animal Husbandry Department has launched several pilot projects in One Health in India. And finally, it has culminated in the establishment of National One Health Mission. It was in 2022 under the Prime Minister's Science, Technology and Innovation Advisory Council formed the National One Health Mission. And as a part of it, National Institute for One Health Nagpur, that is being established in Maharashtra. So the integrated framework of National One Health Mission was a cross-ministerial effort that brought together 13 different ministries and departments in India, including the Indian Council of Medical Research, Department of Animal Husbandry, Department of Pharmaceuticals, Biotechnology, Science and Technology, the Defense Research and Development Organization, CSIR, ICAR, National Disaster Management Authority, NCDC, and Ministry of Health, Ministry of Environment. All these different ministries who are working in their own One Health effort came under the single umbrella of National One Health Mission. So the mission, it was aimed to coordinate overall pandemic preparedness and integrate the control of animal, human, and plant sectors. It was supposed to establish early warning systems built on the integrated surveillance system and the response readiness for endemic, emerging, epidemic, or pandemic threats, and to address a targeted research and development to develop medical countermeasures like vaccines, diagnostics, and therapeutics, and to bring the readiness in terms of clinical care, streamline the data and information. So it was also implementing a integrated disease surveillance across different animal human interfaces in India, and to build a robust outbreak investigation mechanism in India. And as a part of National One Health Mission, there are a lot of attempts are going to to make a network of BSL-3 laboratories in India. That was like under different ministries and departments, so that like whenever there is a pandemic threat that originates in any state in India, we can respond in no time, so that the nearest BSL-3 laboratory with a harmonized laboratory practices and SOPs will respond to it, and we will have a robust mechanism to identify and prevent pandemic threats. So all these laboratories, the BSL-3 network, are equipped to handle human and animal and environmental samples and to diagnose different human and animal pathogens that can be of a pandemic proportion. And there was a national mock drill that has happened in 2024, where a pandemic threat or incident was created. It was a staged one. The purpose of the mock drill was to identify and recognize the gaps that exist in responding to a pandemic threat. So, an exercise was conducted in Rajasthan where a H5N1 outbreak was like a patient was admitted to the local hospital, and how our health system are responding to such a threat, how different departments or different sectors are involved in such a response. So it was evaluated, and it led to a formation of national joint outbreak response team, which has experts from human, animal, and environmental sectors, so that when actually a novel pathogen or a re-emerging pathogen will come in any of our states, we have a robust mechanism, we have a multidisciplinary team to address and respond to the pandemic threat. And under the National One Health Mission, so it's like it's very important that we have a knowledge about what's circulating in the community in case of different respiratory or encephalitis or acute diarrhea, different symptoms, what is actually happening in the community. So there was a launch of a syndromic surveillance network for respiratory viruses along 73 sites in India that can capture the circulating respiratory viruses and other pathogens, and also it can lead to the early detection of any novel pathogen that can arise in India. And we have already established a syndromic surveillance for acute encephalitis syndrome. So as you know, okay. So as you know, like in case of encephalitis, there are a lot of cases which are not diagnosed. I think, can everyone mute?
Participants, please mute. Please mute your phone.
Yeah, so there is a syndromic surveillance network is established for identifying the acute encephalitis syndrome all over India. And we are also establishing a acute febrile illness syndrome network in 38 sites, acute diarrheal disease sites at 20 sites in India. So we can know the landscape of pathogens that's circulating in the community in our country. And under the National One Health Mission, antimicrobial resistant monitoring in hospital wastewater, it's also being established in 60 VRDLs across the country, so that we can identify what's the status of antimicrobial resistance that's happening in the hospital settings, what's happening in the environment, so that we can predict and respond to the threat of AMR. And it's about when we talk about the diagnosis of infections, we know that we have several lacunas in the molecular platforms that are available for detection of pathogens. So it's very important that we should adopt a metagenomics strategy, so that we can capture everything what's there in the environmental sample or a human clinical sample. So metagenomic hubs are being established in India across the 4 important labs that are working in the metagenomics and genomics. And we are also building surveillance models in different kind of animal-human interfaces. A multicentric study is already launched last year, where we are studying the animal, bird, and environmental samples from different bird sanctuaries and wetlands in India, currently it's established in Sikkim, Maharashtra, and Tamil Nadu, where there is like a year throughout the year we are collecting samples from migratory birds. We are also collecting samples from the people who are working in the bird sanctuaries, the inhabitants of the nearby areas, to for early detection of spillovers and to know what's happening in case of the interface that exists in a bird sanctuary. So this project is also will be expanded to more sites in the future. And we are also established a surveillance system in slaughterhouses. A surveillance system in different zoological parks in India is also under it may be launched within a month in India in around 6 different zoos in India. So, as we talk about National One Health Mission, so the National Institute of One Health Nagpur, it will be serving as an institutional anchor for all the coordinated activities of National One Health Mission. It was inaugurated by the Home Prime Minister in 2022, and it is being established with a BSL-3, BSL-4, animal BSL-3 laboratories, which will be involved in training in biosecurity and biosafety practices across sectors in India. And it will be coordinating the pandemic preparedness and outbreak investigation activities throughout the country. So as a part of the National One Health Mission, there was a National One Health Mission Assembly that was organized in the last month in Bharat Mandapam in New Delhi, where there were like 3,000 participants from 14 different ministries, different state health and animal husbandry departments, environmental and wildlife sciences, and expert from national and international community was part of the assembly. And there was a enthusiastic participation from different sectors at the national and state level. So it gives a lot of hope that we are moving very fast in case of pandemic preparedness and in the One Health landscape in India. That's about. Okay, thank you.
Thank you, Dr. Arun, for your ground zero reporting of One Health Center about India's preparedness and our activities. We can have the discussions after the whole panel is over.
Yeah, sure.
Hope that you will be with us.
Sure.
Okay.
Sure. Thank you.
Thank you, sir. If there are any queries, we'll address it at the end of the session. Next, we have with us Dr. Mohammed, a research fellow at the University of Melbourne, Australia, specializing in One Health, climate change, and food security. He previously served as an assistant professor at the Kerala Veterinary and Animal Sciences University and holds a PhD in animal breeding, genetics, and biostatistics. Dr. Mohammed has an impressive academic record with 35 peer-reviewed journal publications, 2 book chapters, and over 60 popular science articles featured in leading newspapers and magazines. His contributions to animal sciences, sustainability, and genomics have earned him global recognition, including the Australian Global Talent Visa and fellowship from Rothamsted International UK and Crawford Fund, Australia. He also actively contributes to academic and professional bodies such as Australian Association of Animal Sciences and has served on the Board of Studies at Kerala University of Health Sciences. Today, he will talk on the topic Zoonotic Spills, Pathways and Prevention. Now I invite Dr. Jayakrishnan to kindly present our speaker and introduce the topic to our audience. Jayakrishnan, sir, please.
As mentioned by the first speaker, the main threats of emerging infectious diseases are This is due to zoonotic diseases. Historically, pandemics have happened due to zoonotic disease. Examples, in old age, it was plague, as well as in the modern age, it was due to COVID-19. It was proven that historically, TB reached from bovine TB when cattle were domesticated 10,000 to 15,000 years back, and human measles were derived from animals by accidental spills. Currently, in 21st century, in 2003, emerging disease like SARS coronavirus originated at China. In 2009, H1N1 at Mexico. 2012, Middle East respiratory virus at Jeddah. And last 2020, COVID-19 at Wuhan, China. As well as, along with the emerging diseases, re-emerging diseases like Ebola in Africa in 2014, monkeypox in European Union 2022 were all zoonotic in origin. What are the driving forces for zoonotic spells? Why they occur in increased frequencies now? What are the pathways of spillover? What should be our future action plans? So this session will enlighten us on this area. I invite our speaker of today, Dr. Mohammad Elayed Meetal, for enlightening us for SNS speaker. Over to Dr. Mohammad. So, Dr. Mohammad?
Yes.
Sir.
Am I audible?
You are audible. You are audible. You are audible.
Yeah. Okay.
But slides can be changed as presentation mode. It is now in the slide mode. It can change to presentation mode.
Thank you. Is it okay now?
Yes, okay.
Okay. Thank you very much. Thank you, Nawaz sir, and the whole KMCT team for providing the opportunity to collaborate with you for the last couple of years in academic domain and also in the research areas. For today's presentation, we'll be just discussing zoonotic spills and its pathways and prevention. We'll move on to the presentation. I have just compiled the zoonotic spills in the last 35 years, in which most of these zoonotic spills occurred in Asian countries, except a couple of zoonotic spills that happened in Australia and elsewhere. So it was back in 1994 that there was a hentravirus infection, zoonotic infection in Australia. And in 2015, there was Zika virus infection. In exception to these couple of zoonotic spills, most of those zoonotic spills happened in Asian and African countries. So it's of highly interest for both the public and policymakers to intervene in this area. Yes. So basically, being an academician and a teacher, so first let me explain you what is actually zoonotic spill or spillover. It simply means that a pathogen that is causing a disease in animals, or just without causing a disease, a pathogen is there in the animal population, such as a virus, bacterium, or a parasite, jumps from an animal host to humans, causing infection in people. So that is what is called a zoonotic spill or spillover. The spillover happens when ecological, biological, and social conditions allow a pathogen circulating in animals to cross species barriers and establish transmission in humans. So in simpler terms, I will explain this phenomena with the COVID-19. For example, how COVID-19 spillover can happen in the current scenario. So in such cases, what happened is, for SARS-CoV-2 virus, the receptors are ACE2 receptor, angiotensin converting enzyme 2 receptor. And this receptor is responsible for inviting the pathogen to a host and accommodating that pathogen and providing all the facilities for that pathogen to enter and multiply in the body. So this spillover can happen in two ways. One, the pathogen SARS-CoV-2 virus attains virulence to attach to ACE2 receptor in humans. That is one possibility. Or otherwise, just the reverse can also happen, wherein some mutations happen in the ACE2 receptor loci in human species. And historically, our ACE2 receptors are resistant to SARS-CoV-2 viral proteins, viral antigens. But due to some mutations, ACE2 become receptive to SARS-CoV-2. So that is the basic mechanism, genetic mechanism that is happening in spillover cases. Let us see that in detail. Yeah, this being the mechanism, why it is important? Zoonotic spill is important. All the governments all over the world and policymakers and public are alert now, because about 60 to 70% of emerging infectious diseases in humans are zoonotic. And previously we thought zoonotic spill happens from animals to humans, but the reverse also happened, which is called a reverse spill from humans to animals in a very high alarming rate in recent times. For example, as Arun already mentioned, the SARS-CoV-2 virus causing COVID-19 backspilled into more than 30 species of animals, including domestic and wild animals. So it's a real concern in this. So what are the drivers, or what are the factors that may increase these chances? One is land use change and deforestation. More and more land is being used for urbanization due to an increased population and also increased lifestyle changes. Then the second reason is industrial livestock farming, because livestock as a whole, traditionally they were reared under traditional conditions, but nowadays industrial livestock farming has changed livestock nutrition, livestock management, everything. So genetically also, livestock genome is changing, mutations are happening rapidly, so that receptors are more and more becoming receptive to more and more viruses, bacteria, and other pathogens. Wildlife trade and wet markets, and also climate change altering the wildlife movement. So the spillover risk is rising as human-animal interfaces expand. Zoonotic spillover is shaped by interactions between three major host groups, as we have already seen: wild animals, domestic animals, and humans. So this is usually called as the spillover triad. for zoonotic spillover. This forms the triad. For humans, in most of the cases, they are final or incidental host, sometimes becoming new reservoirs. So humans become reservoirs and they are becoming source of infection to other.
The concentration of oxygen is 104 and concentration of carbon dioxide is.
Okay. Then domestic animals act. Okay. Am I still audible, sir?
Yes, sir.
Okay, thank you. Domestic animals act as an intermediate host or amplifiers. For example, pigs for Nipah virus infections, pig act as an amplifier host. Camels for MERS, poultry for avian influenza. So these are important amplifying hosts for major spillover zoonotic diseases, which caused major havoc in different countries and different places. Wildlife reservoirs where pathogens evolved naturally, bats, rodents, and primates. They are actually the reservoirs wherein they cause no diseases in those animals, no symptoms in those animals, but they carry the organism. They happily live together. But due to some reasons, these viruses or pathogens escape from this wildlife, enter into domestic animals, and find a good pathway to reach into humans, causing the whole trouble. Now let us see how this happened. What is the mechanistic process, biological process by which a zoonotic spill happened? That means some viruses that is happily living in wild animals like bats enter into domestic animals like pigs, poultry, or camel, or whatever it may be, the intermediate host, and reaching to the humans. To explain you a little bit with examples, say for example, almost all the zoonotic spills happened through the intermediary pathway with the wild animals, then domestic animals, and ultimately to humans. In case of Nipah virus, bats were the origin. Through pigs, it entered into humans. H5N1 influenza, avian influenza, the source was waterfowls, and through poultry, it reached to humans. MERS-CoV, again, source was bats, and then through camels, it entered into humans. Ebola, again, bats, through primates, or maybe directly to humans, both evidence are there, and they reached humans. So this was the pathway zoonotic spill happened. Each step in this triad allows pathogens to encounter new environments and evolve new infection strategies. That is what happened. I'll tell you how this happened. So we will see spectrum of evolution of spillover mechanisms. The interaction between host, so for example, wildlife, livestock, and humans, and pathogens exist along a spectrum of host response. That means there are three mechanisms. One is resistance, second one is tolerance, and the third one is resilience. These responses shape whether a pathogen remains confined to an animal host or evolves the capacity to spill over into new species, including humans. So as molecular geneticists, what we do is we understand the mechanism of resistance, tolerance, and resilience of these pathogens in wildlife, livestock, and humans. Thereby, we could understand which pathogen is going to enter into next species level, next spectrum in the continuum, and which is going to become an epidemic or a pandemic according to the mutations or changes that is happening either in the parasite, pathogen, or in the host species. So see, what is resistance actually? Resistance means simply the organism or a host. Say, for example, humans. In humans, some of those diseases will not affect us. Why? Because we are resistant. Resistance means ability of an animal or a host or a human to prevent entry, replication, or survival of a pathogen. That means at no cost, that organism, that species will not allow the entry of the organism. Even if the organism enters, it will not allow the replication, or simply the pathogens will not survive in that species. In that case, that species is totally resistant to that particular pathogen. Example, you may have load of examples. One example I will quote: bat species, which evolved strong antiviral defenses that prevent coronaviruses from causing diseases. That is why they act as reservoir host. That means the receptors are not working, or their immune mechanisms are so strong against the pathogenic antigen that those pathogens will not have an entry into the organism. Then the second level of that continuum is tolerance. In this case, the species will be, whether it is human or an animal, is unable to prevent the entry of the organism, but it will allow pathogen entry because it can't prevent the entry. It will also allow replication, but if the animal is tolerant, in that case, it will limit the disease symptoms. Bats tolerate Nipah virus infections with minimal clinical disease, allowing long-term viral shedding. Bats are not resistant to Nipah virus, but they are only tolerant. That is why any cases of mild stress will enhance the viral shedding in case of Nipah virus. That will lead to a sort of a zoonotic spill in vulnerable areas where there are bat and human interactions. Resilience is the last and the most important spectrum in this continuum, wherein host will never have the facility, have the capacity to resist the entry of the organism. Organism will enter freely, and organism will multiply freely. It cannot prevent that. The immune system is so weak against that pathogen. But again, it will cause a measurable disease. And according to the level of immunity in the individual animal, not the species, because species is not immune. But according to the individual immunity, the individual will respond to the disease, to the pathogen. For example, it may be reduced performance, showing some symptoms, or at the extreme stage, it will lead to mortality. One classical example in this case is with the humans, where in Lassa fever, it shows only mild or moderate illness. And most of the humans will recover, but some of the unfortunate, unlucky individuals, they may die. So these are the three continuum of spectrum in which zoonotic spill happens, and this is the molecular mechanisms happening. This will be of interest to those researchers and students who are really interested to work on this area. I have represented this in a graphical representation with 5 diseases. Say, for example, influenza, Nipah, Ebola, And again, these diseases, how does spillover happens in this disease? We'll see the first in wildlife. Generally, wildlife is resistant. So they will accommodate these organisms, never allow to go outside from their body. Happy, both of them are happy. And we are also happy. No spillover, no disease for us. But somehow there happens something either in the organism, in the pathogen, some mutations happen in the pathogen, or something happens to the host. So for example, bats become less tolerant, or they lose their resistance and they start showing symptoms. In such cases, what happens? It is the trigger for spillover. So nowadays, in most of the zoonotic diseases, we have identified spillover mutations in both pathogens and also in the wild animal species. So by mass screening of pathogens and wildlife, nowadays it is possible to predict, is there any possibility, or even in the coming years, within 10 years or within 20 years, because mutations have a lifespan and it's a continuous process. So we can predict within 10 years what is going to happen for this particular gene that is causing spillover in pathogen or in wild species of animal. So wildlife loses its resistance, and then it becomes less tolerant and starts showing symptoms, and it starts shedding viruses. And in this case, the livestock or any other domestic animal, the pathogen directly enters into these animals. And in most of the cases, sometimes domestic animals also may be tolerant, but sometimes it may show some sort of disease symptoms, but mostly unnoticed. And in this case, what happens is, as we discussed earlier, some of those domestic animals act as amplifying host. That means both things happen. I mean, the pathogen gains more virulence and the organism, the host, is becoming less resistant. So in such cases, it may show some symptoms in animals. Again, when our animal-human contacts happen, that will result in diseases in humans, resulting in a zoonotic spill. So this is actually the mechanism that's happening. Now, it's an interesting area in which we have to intervene, and we are working on. Future research into zoonotic spillover will critically depend on understanding the evolution and dynamics of host responses, specifically resistance, tolerance, and resilience across wildlife, domestic animals, and humans. This is a huge area of study. Future research should aim to map host pathogen networks in wildlife, domestic animals, and humans. Then quantify the evolutionary pressures on host species that favor specific immunological responses. I mean, when a simple and very friendly influenza virus turns into a hostile symptoms causing virus, we have to make it sure that it does not go beyond that level to become a pandemic or a zoonotic spill. Then model spillover probability for various ecological scenarios. Now, especially in developing countries, this is a really worrying situation wherein we have different ecological systems. We have more access, more contact with the wildlife. And also our farming system is more towards rural areas. so that human, farm animal, wildlife, these interactions are much common. So in order to make a prediction model for future spillover, we may really need to work hard, especially in the Asian and African context. Then integrate longitudinal surveillance data to evaluate changes in host susceptibility. Remember, in 2025, if you find it is perfectly okay, we are not expecting any zoonotic spillover in 25 or 26, but mutations are always happening on a time scale. So we should definitely target a 10-year, 20-year time frame for this sort of research. Otherwise, it will be very disastrous to see this sort of zoonotic spillovers in the coming years, if not in the near future. Then create predictive frameworks for early detection of emerging pathogens. So even if a pathogen is totally harmless, totally friendly nowadays, but we can really predict at what stage, in which period it can go and cause a zoonotic spill or by increasing its virulence. And what are all the host, pathogen, and environmental factors, these three factors that may act together to become a pandemic or a zoonotic spill. Thank you. Those who are really interested to know further, you can go for these references, and also see the in-depth mechanisms. I have only given a superficial outline of those who are really interested to understand those mechanisms of zoonotic spills, and those who really are interested in an in-depth study can go through these references. Thank you. Thank you very much.
Thank you, Dr. Mohammed, on throwing light on the pathways of spillovers. The questions can be answered later. Now, we can go to the next speaker.
Thank you, sir. Our next scientific session is by Dr. Deep Jyoti Buyan, Senior Research Fellow at Western Sydney University, where he also serves as the Global Challenge Lead and SDG 3 Research Cluster Lead with UNESCO and the International Association of Universities. Dr. Buyan leads the Gut Biotics Lab at the NICM Health Research Institute, a pioneering research center whose work has significantly advanced the global understanding of gut health, particularly the emerging role of postbiotics in enhancing chemotherapy outcomes and shaping future clinical and translational research. He has an extensive research, and his groundbreaking work on postbiotics has garnered wide international attention, reaching over 39 million readers and earning multiple recognitions, including the 2023 Marcus Blackmore Outstanding Contribution to Research Award. Beyond academia, he contributes to advisory boards and has facilitated the commercialization of functional food products both in Australia and worldwide. Today, he will be speaking on the topic postbiotics, their role in gut health. May I now invite Dr. Jayakrishnan to kindly present our speaker and introduce the topic to our audience. Over to Jayakrishnan, sir.
The previous talk was on zoonotic disease. In the planet Earth, we Homo sapiens are living in an environment which perceived mainly as external. But at the same time, we Homo sapiens are having an internal environment too, or an ecosystem, which is called microenvironment in our body, which are diverse also, especially over the skin outside and the gut inside. Of this microenvironment, the gut biology is important. Trillions of microorganisms are living in our gut: bacteria, fungi, and viruses. All of you know now the importance of gut-brain axis. How to keep balance in the gut microbiome biodiversity? What are the role of fermented foods in maintaining human ecology? What are the role of postbiotics? Future directions of drug research in these areas. Dr. Buyan, who leads at Gut Biotics Lab, will throw light on it. Over to Dr. Deep Jyoti Buyan on the topic postbiotics and the role of in gut health. Please, Dr. Buyan, you can start now.
Thank you. I'll share my screen. Is my screen visible to everyone?
Yes, your screen is visible as well as you are audible also. You can
Okay,
start.
great. Thank you. Thank you so much to, first of all, Dr. Santosh for the kind invitation and Dr. Nawaz as well. And thanks to Dr. T. Jayakrishnan for the very long and kind introduction as well. I would like to show you this particular image, which is an indigenous artwork from Australia. And this was done at our university a couple of years ago. And this basically captures the, today's core theme of One Health in one picture, showing humans, plants, microorganisms in one picture. So I'm going to shift the gears a little bit in today's presentation. We have heard, amazing speakers talking about zoonotic diseases and antimicrobial resistance. My presentation will be mostly inward, so looking at what happens inside the body and what are the things we could do in terms of preventing diseases, including infectious diseases, and other lifestyle-related diseases as well. So, all right. So this is my presentation. So I'm going to talk about a new or an emerging concept of postbiotics and how that relates to One Health and gut health particularly. So would like to begin with these stats, which are quite daunting in a way. So if you look at these stats, we can see that about 94% Indians do not eat their daily vegetable intake. And same goes for Australia. About 92% of Australians don't meet their daily recommended vegetable intake. I'll tell you why I'm talking about nutrition here in a little bit of time. So basically, what happens is that when it comes to poor nutrition, it can lead to different diseases. It can contribute to infectious diseases as well because of the link with the immunity. So India spend about, I'm not going to actually, tell you how much money. So you can see there, it's a lot of money. I think it's probably 9 million crores, or 900 crores rupees on their healthcare system, and about 50% of that come from out of pocket. Australia is similarly quite expensive when it comes to the healthcare system. It is about 195 billion every year. So we think, I might be biased because I have a research group on gut microbiome, but we think that our nutrition and the microbiome has a big role to play there. So I'm not going to talk about COVID-19. I think Dr. Arun has beautifully explained all the scenarios that happened during COVID-19 and how the world came to a stop. But also, it kind of outlines the impact of a small virus that can have on humanity and overall the world, which basically captures the concept of One Health in a beautiful way. So I'm talking about bacteria here. I won't be talking about viruses. So I would like you to show you a few numbers, and I want you to make a guess of what percentage of bacteria is present in our bodies. So basically, if you count all the cells, human cells and bacterial cells, how much of that would be humans and how much of that would be bacteria? I'll give you a few options. Please feel free to put your answers on the chat box, and I will tell you the answer shortly anyway. But yeah, please feel free to have a guess. So what percentage of our bacteria, our body is bacteria? So these are your options. So you have 10%, 27%, and 57%. So please feel free to put some numbers in the chat, and I'll tell you the answers shortly. Okay, let's see. 57. Yeah, I can 57, 57. Okay, 27. All right. Okay, 57. Yeah. Okay. So I think I'm talking to an audience that know microbiology. So I think you got most of it correctly, because about 57% of the cells, if you count human cells and bacterial cells on our body, you'll find that 57 represent the bacterial cells. So that actually shows you that we are probably more bacteria or fungi and viruses and archaea, it's essentially the microbiome, than human. And that is why we need to pay attention to the topic of the gut microbiome or microbiome in general. All right. So when you go to a doctor, there are three things the doctor is going to mention it to you if you want to maintain a good health. So those things are have good nutrition. I told you about the importance of nutrition previously, but yeah, nutrition is key. Then you need to exercise daily, about 20 minutes of walk every day, and you need to sleep regularly. But what happens when you do all these things to the point, but even then you don't see an ideal health outcome? So in that case, I think our microbiome has a big role to play. But as I said previously, I might be biased because I am a researcher in the gut microbiome field. But I do feel that there is quite a lot of credibility when it comes to the research that has been done on the gut microbiome so far. So our concept, or the research concept, is quite simple, and it is inspired by this age-old saying from Hippocrates, who said that all disease begins in the gut. So we think that our health begins in the gut as well. But then again, when it comes to One Health, everything is influenced. So can influence our gut microbiome, including our diet, the environment, the medicines that you take, your lifestyle, and all that. So the gut microbiome is a very dynamic environment, and things are constantly evolving. But having said that, to study gut microbiome, we think that our poo can tell a lot of things about our health in terms of what kind of diseases we are going to get in the future. So it can predict as well. So our poo, although it is excretion excretory agent, we can actually use it for our advantage when it comes to studying the gut microbiome. So this is where our scientific curiosities actually begin. So we want to understand complex cellular processes in diseases like cancer and cardiovascular diseases, and also study the gut microbiota and look at how the food and nutrition angle fit into that equation. And that's when I started this research group at Western Sydney University back in 2021 during COVID time, actually. And since then, that group has grown quite a bit. So as you can see here, our group currently has about 13 students, and these are the amazing students that do all the hard work that Are we talking about the microbiome, and what we're talking about inward, what is inside our body and how that influences our health? Because of many reasons. Because these are, as I said, these are the trillions of microorganisms that are present in the gut, and because of their importance, as I said, 57% is bacteria. So now they are categorized as a separate organ in the body. Although it is considered an invisible organ, its significance in health and disease is immense. And when it comes to food itself, so about 90 to 95% of antioxidants that we eat through our fruits and veggies, those are metabolized by the gut microbiota. And it also helps with protection from pathogen. That's when the concept of One Health comes in, where it helps modulating the immune system, and also influence the response, how you respond to different drugs. Production of neurotransmitters. I think Dr. Jai Nutrition has already mentioned about the gut-brain axis, and that is one key concept that is gaining a lot of attention these days. And of course, it helps with the metabolism of food and other things that you consume orally, and also maintains the gut barrier integrity. All right. So I will specifically talk about postbiotics here. I'm not going to talk a lot about the communities, but postbiotics are essentially the byproduct of the gut microbiota. So basically, when you eat prebiotic and probiotic foods, your gut microbial communities are going to metabolize that and produce these amazing molecules called postbiotics. And now the whole world of microbiology, especially the gut microbiome research field, is moving towards postbiotics because we are more interested to learn about the chemical signatures of the gut microbiota rather than the microbial communities, or in addition to the microbial communities. Because we think that most of the work, most of the impact that we see of the gut microbiota on health is mediated through postbiotics. So to inform this concept, so it's an emerging concept. I wrote a few articles for the news channels here in Australia as well as in India, like Indian Express and BBC as well, BBC News, to explain the concept of postbiotics and how it is important in today's scenario when we talk about the microbiota. So the concept is, although now getting a lot of attention, it is not necessarily new. It started back in 1986, and there's a number of postbiotics that have been studied so far. And there's a paper that kind of outlines the role of postbiotics in COVID-19, especially to manage post-COVID-19 complications. Please feel free to have a look. And I found this particular paper is quite interesting. And it talks about a number of postbiotics, including short-chain fatty acids and lipopolysaccharides and bacteriocins, which are produced by the gut microbiota inside the body, and how they can have different types of applications when it comes to the pharmaceutical industry as well as the food industry, in terms of having antimicrobial activity, in terms of having obesity activity, anti-diabetic activity, anti-cancer activity, which is one of our expertise as well. So it basically captures all those activities quite nicely. So in terms of the clinical evidence, so there has been a number of papers that have been recently published looking at so a number of clinical trials, I would say, as well as meta-analysis that talked about the efficacy of postbiotics, especially in gut health and infectious diseases and diarrhea. So having said that, like, not all postbiotics are good. There are bad postbiotics as well. So what happens is that when you consume a bad diet, let's say a hard diet that is high in fat and meat, red meat, for example, especially Western diet, and now that India is considered the diabetic capital in the world, and as well as it's highly prevalent to have Western diet in India with McDonald's, KFCs, and all that. There are chances that that can lead to modulation of your gut microbiota in a negative way, and that can actually lead to formation of toxic metabolites, which are considered bad postbiotics, such as TMAO, which can lead to diseases like heart failure and atherosclerosis. In terms of our research, so in relation to gut health, so we have done a number of studies, commercially available products, to inform the development of new formulation, prebiotic and probiotic formulation, as well as postbiotic formulations. And here are some of the studies that we have done where we found that a number of prebiotic formulation. This was funded through an industry partner. We were able to see that the prebiotic formulation was able to modulate the microbiome, and it was quite potent in terms of against pathogens such as E. coli, and it has very strong antiviral actions against H-CoV-229E, which is a model organism for COVID-19. And also we found that even though the product was going through the gastric system, the activity was intact even after gastric digestion. So that was quite a promising finding in terms of that particular formula that we developed in partnership with industry. In relation to cancer, we have done quite a lot of work in that space. looking at how postbiotics, which are produced by the gut microbiota, can help in the prevention and treatment of cancer. And we also were able to quantify how they interact with standard chemotherapy in a positive way. So essentially what we did is that we combined postbiotics with standard chemotherapy, and we were able to see quite a lot of increase in activity by several fold of the chemotherapy within the combination. So basically, when you combine postbiotics with standard chemotherapy, certain postbiotics, it was able to kill the cancer cells quite effectively, while it was quite inactive or ineffective towards the normal healthy cells. So that's a very good indication of its therapeutic index and how it can be implemented in cancer therapy in the future. So this was done on gastric adenocarcinoma cells, and you can see from the images here that the combination was able to increase the level of apoptosis in the cancer cell, which is a good indication for any sort of anti-cancer drug. In relation to nutrition research as well, so we found that when the short chain fatty acid combination, we're also able to improve the health of the gut cells. So which means that, when you're giving this in terms as a therapeutic agent in combination with doxorubicin or other anti-cancer drugs, it was able to kill the cancer cells, but it was actually it was able to heal the gut at a cellular level. And similarly, other postbiotics were also studied, like niacin and urotin B, which are also found. These are produced by the microbiota when we consume a certain food. We've done quite a lot of work in the Australian native fruits and vegetable space as well, looking at the bioactivity and how they can be effective in treating infectious diseases and cancer. And what we found is that, mostly with all the foods, like for example, fruits and vegetables that we consume it mostly for fiber and antioxidants. But the bad thing is that only about 5 to 10% of those polyphenols are bioavailable, so that are actually absorbed by the small intestine. Remaining 90 to 95% actually reach the colon, and then it undergoes fermentation by the gut microbiota. So essentially, if you do not have an ideal gut microbiota, you're not going to get the most benefit out of your food. And that kind of links with my previous slide where I said that nutrition, the missing link to nutrition is or might be the gut microbiota. So as you can see that most of the nutrients are metabolized by the gut microbiota, and after that, your body is able to absorb them effectively. So this is why we actually design a number of studies looking at how we can do this step outside the body. So basically, we are using lactic acid bacteria, which are probiotic gut bacteria that are considered healthy. So we use that to ferment a number of Australian native and fruits and vegetables, and to improve the bioavailability of their antioxidants so that it can confer better health benefits. So these are a number of projects that we are currently doing. We are also looking at 3-dimensional organoids. So basically, we can develop 3-dimensional organoid system for the intestine and look at how that can be used for, to study bioavailability of postbiotics, as well as to understand the toxicity and interaction with the tumor microenvironment, as well as the gut microbiota. And of course, the interaction of postbiotics with standard drug as well. But yeah, there are a lot of challenges. So if you look at the current literature, you will see that a lot of postbiotic research has been done on 2 bacterial strains only. So mostly like Lactobacillus and Bifidobacterium. And there are some new ones like Akkermansia species that are evolving, and people have started to pay attention to that as well. But yeah, there's quite a lot of work that need to be done in that area. Also defining what is good and what is bad gut microbiota is quite difficult, because as we are talking about trillions of microorganisms and thousands of metabolites, it's very hard to characterize that what is good and what is bad. So we have to look at one thing at a time, or to understand their in-depth effect on health. Yeah, so as I said, it is a very complicated area, but quite challenging and quite fascinating. So we definitely need more randomized and placebo-controlled trials to understand how postbiotics can be helpful in health. So this is the lab that I lead. It's called the Gut Biotics Lab at Nicum Health Research Institute at Western Sydney University. Please feel free to have a look if you are interested in our research. So this QR code you can see here. If you scan that, it will directly take you to the website, and you can have a look at all the papers and projects that we currently have. And these are some of our academic collaborations. We have been quite lucky to have a very extensive international collaborations, and we have published with all these collaborators so far. So, and we are actively working as well. Some of our papers from the lab, so you can see that there are a lot of work in the gut microbiota and postbiotic space, as well as in cancer and food science. So yeah, if anything tickles your fancy, please feel free to reach out to us in terms of doing more work together. So the food for thought is quite simple, I think, especially I'm talking to an audience from South India, and I think you guys are amazing when it comes to your food habits. But the rest of India can probably learn a bit from you in terms of your food habits and nutrition. I think fermented foods is probably the key when it comes to improving your beneficial gut microbiota inside the gut, which will eventually help you with fighting infectious diseases and as well as improve your immunity. Also pay a lot of attention to fiber. So eat fiber-rich foods, legumes, and nuts. As I said, this presentation is slightly deviated from the One Health theme, but I want to emphasize that nutrition is key here, and we need to look inside before we can tackle the problems outside. So yeah, thank you so much for listening, and I'm slightly over time. I'm really sorry for that. But yeah, happy to take questions later on. Thank you.
Thank you, Dr. Deep Jyoti Bhuyan. Your topic area is a new area, which most of the participants are not familiar. So inside, you reveal that not outside, but inside the human body exists a large forest. So the One Health, you have to concentrate on not only on the external forest, but also internal forest. Thank you so for enlightening us in the new area.
Thank you.
Okay. Please remain here for the end of the discussions. That will be done after the whole topic is completed.
Thank you, sir. KMCT is pleased to express our gratitude to Dr. Deep Bhuyan from Western Sydney University for his insightful talk. We're also delighted to share that this collaboration has been strengthened by our recent MOU with Western Sydney University, and KMCT look forward to many more opportunities for academic exchange and growth. Thank you so much, sir. And now we move on to our next scientific session. The next session would be by Dr. Minu Divakaran, a distinguished professor of plant sciences with over 30 years of experience in research and teaching. Presently, Dr. Minu is the Director of Research and Consultancy, Providence Women's College, Kozhikode. Her expertise spans genetics and plant breeding, as well as plant biotechnology. Her work has earned both national and international recognition, including her prestigious induction as a Fellow of the Linnean Society of London. She has also been invited for research collaborations in Mexico and France, reflecting the global impact of her scholarship. Currently, she serves as a NABET Functional Area Expert in Ecology and Biodiversity. Her academic output is extensive, with over 100 research publications, contributions to international book chapters, and leadership of externally funded research projects, all of which highlight her sustained commitment to advancing plant science and research. Today's session will feature her insights on the topic, Emerging Frontiers in Plant Diversity. May I now invite Dr. Jayakrishnan to kindly present our speaker and introduce the topic to our audience. Over to Dr. Jayakrishnan.
The previous topic by Dr. D. Jyoti Biyan was on about the micro environment inside the body. Now we can move to the macro environment. One of the prime requests for human or animal health is ecological balance, which demands ecological diversity. An ecosystem mainly consists living animals and plants. Many of One Health problems are due to disturbance in plant ecosystem or diversity. So maintaining plant diversity is important. The first antibiotic discovered by Alexander Fleming in 1928, penicillin, was derived from plant sources, fungi. So in other words, plants can be considered as health resources, or plants give curative solutions for our One Health problems. So how biotechnology can be utilized in future, Dr. Minu will elaborate these points. Over to Dr. Minu Divyagaran for the enlightening session on emerging frontiers in plant diversity. Please, madam, you can start.
Thank you. Thank you, sir. Delegates and all professionals who are joining this conference focused on the One Health concept. Thank you for the warm welcome and also for the honor of making a plant science researcher part of this conference. The sign that plant sciences have been included in this conference itself shows the role of plants in human health as well as the environment. I would like to discuss or share my experiences of working with plant diversity and the different emerging frontiers and tools that are being used to improve plants. My world is basically rooted in genetics, plant breeding, and plant biotechnology, all focused towards the improvement of plants. Plants have remained the unseen foundation at large, but all of us know the pyramid of energy where the foundation is plants. And the most recent pandemic, COVID-19, has also taught us the importance of food, primarily plant-based food, and the need for oxygen, which were the primary requirements of all humankind during the period. The rest of the living components in this ecosystem are dependent on this foundation, which remains unseen yet directly relates to the human health, animal health, and the environmental health. And therefore, the health of the plant is of utmost importance. The plant health pyramid, which has 4 levels, the first and second, or the base, the 2 levels at the base, largely are about the passive immunity, while the 3rd and 4th levels, or the upper levels, are related to the active immunity. The first level depends on proper and complete photosynthesis, which also indicates the beginning of natural resistance, development of natural resistance. The second level, where complete protein synthesis needs to happen, all the nitrates have to be converted to amino acids and proteins, failing which it would affect the health of the plant and the ecosystem. The first and second levels, if goes perfectly well, leads to the 3rd level, that is the increased lipid synthesis, which is directly related to the formation of cell membranes, etc., making the plant tissue or plant hard to be infected or affected by other pathogens. And the 4th level is the production of plant secondary metabolites, which is directly related to the advanced immune system. It gets activated by the production of plant secondary metabolites. So it is needless to say how the level 1, or the happening of a complete photosynthetic pathway, is directly related to the advanced immune system in plants. And healthy plants naturally become resistant to diseases and pests. If we look at the current scenario that is around us, we can see a non-expansive environment or ecosystem or the natural resources versus an ever-expanding population, such that there is a lot of pressure on the natural resources. There isn't enough food for the ever-increasing population. That is the current scenario of our globe. And agriculture practices that are being undertaken directly or indirectly are associating with increasing the AMR risk, which is a global threat now. As plant sciences people or researchers, our job is to see that prevention happens before there is a need to cure diseases. And that is the reason why we look out for safer food production options. And some of the tools or technologies that are emerging, and I would like to include in today's session, are plant tissue culture, the isolation of protoplast and fusing them with another protoplast of a totally different genera, cryogene banks, metabolite engineering, CRISPR-Cas techniques, genetic engineering, and much more, which are all based on utilizing the plant diversity and for improving crop-specific problems or crop-specific traits. So my session would be primarily a plant-centric view, focusing, of course, on One Health options, the threats, and the solutions. While I was just looking at the past few reports of the FAO and UNICEF, the cover pages always focused on plant-based food when it comes to food security. And one of the biggest threat that the globe faces now is the antimicrobial resistance. I just tried to put in a few examples of plants that we use in our everyday life quite unknowingly. For example, the spices, cloves, and cinnamon, which has the secondary metabolites in them, like eugenol and cinnamaldehyde, respectively. The action that these metabolites has on the cell membrane function and structure leads them to become or leads their antifungal activity, which works against the drug-resistant fungi. Similarly, ginger, which has the metabolite gingerol, and that interferes with drug resistance virus. And the common fruit that all of us use, guava, has hydrosoluble compounds which can actually reduce or inhibit the egg laying in parasites. So it has an anti-parasitic activity against the different drug-resistant parasites. So these are some of the naturally available antifungal, antibacterial, and anti-parasitic activity of the crops that we are using in our daily lives against or to break the antimicrobial resistance. Of course, a lot more of standardization has to happen before it can be used more actively. Now, these features or these secondary metabolites are actually proof of how plant diversity is directly related to the phytochemical diversity available within the plant. They are actually reservoirs of phytochemicals. Some of the studies that we are undertaking in our lab, and my students are working on it, is understanding the potential of neglected or underutilized crops, as we call them. One being euphorbia. All of us would have trampled over it unknowingly. And the extracts from this plant was very promising in that you can see the images here. They are, of course, lab-based experiments. And in Petri dishes, most of the bacterial strains showed an inhibitory zone to these commonly seen bacterial infections or bacterial strains. Similarly, we also tried green synthesis of nanoparticles, especially from long pepper, which we commonly call as tipali or botanically as piper longum, a related species of black pepper. And we found that the nano extracts were very efficient in inhibiting the growth of bacterial isolates. Again, a lab experiment. And these applications are a promise to also addressing the Sustainable Development Goal, especially the Goal 3, which focuses on good health and well-being. So these are actually safe alternatives to the chemically synthesized antibacterial medicine. When we look at all these, the current scenario that the globe faces is that of a food insecure planet that directly leads to unsustainable development. Now, the world population, which is rising at an alarming rate, coupled with the unprecedented climatic changes, all affect the availability of food on the earth. The agriculture production goes down. The land that is currently under agriculture cultivation is also reducing at a drastic rate because, as I mentioned, the natural resources are non-expansive, whereas the population is ever expanding. So the world population itself creates a big challenge on us because providing food for all is a big challenge. Agriculture basically practices monoculture, which also has adverse effects. In this context, I would like to present a technology called plant tissue culture, where the core concept focuses on providing a defined culture media or a nutrient-rich media under absolutely controlled conditions or environment. If we are able to provide these two requirements of living cells, there are a lot we can do to address and elevate the crop-specific problems. For example, the production of secondary metabolites. Conventional method depends on harvesting the entire plants, which is called destructive sampling or making of medicine for the making of medicines. But secondary metabolites can be produced in vitro using this method. Similarly, multiplying plants in large numbers because we are losing a lot of the plant diversity with important genes from the nature or environment. And micropropagation is one such method where you can bring them back to the nature by multiplying them in large numbers under controlled conditions. Then the production of genetic transformants using genetic engineering or even new hybrids with the help of protoplast culture and conserving whatever diversity we have generated in safe repositories like cryogene banks for the posterity. These are some of the tools that I would be showing. And climate change is almost inevitable. Most of us are going through it. It has led to a lot of collapse in the nutritional value of many food crops. And many of the staple crops like rice, wheat, etc., have very low levels of micronutrients. The consequence is that it affects the global health. The impact is that it affects the global health, leading to iron deficiency, that is anemia, and also zinc deficiency, which impairs the human or any life system, immune functions. So the net outcome is that though our plate looks abundant with food, but it is an illusion of nutrition. I was very happy that the previous speaker, Dr. Dev, he ended his presentation with a very interesting note, or rather a message to people that you have to be careful about what you consume. And Sir had included all plant-based products. And in the beginning, Sir had also mentioned that you have to, or Indians and Australians are just taking 94% of their daily dietary requirement, which is quite alarming. So when you resort to these technologies, for example, the golden rice. Rice is a staple diet, but it doesn't supplement other dietary requirements, mostly carbohydrates. So rice was genetically engineered. In this image, you could see the white grains, which is the ordinary rice, and the golden colored grains, which are the genetically modified rice grains. Two genes have been inserted into the genome of rice, and genetic transformation was done two genes which actually are biosynthetic genes for the beta carotene pathway. So the transformed rice started synthesizing beta carotene on its own, and that resulted in this golden yellowish color. So this intake or the consumption of golden rice led to improve the vitamin A deficiency in many of the countries, and also independence from the nutrient supplementation, external supplementation programs. So this was a big relief to the countries which are suffering from vitamin A deficiency to a lethality level. Now, I said as human population increases, the resources are diminishing. They are not proportionate to the growing population requirement. And the plant population as such is reducing. All of us are witness to the loss of green around us by way of human interventions in the name of development, urban development, etc. Now, in the process, what happens is a lot of plant diversity is being lost. And as plant science researchers, it is our responsibility to try to multiply them in large numbers, because many of those species which are being lost are already under the RET category of IUCN, that stands for rare, endangered, and threatened. In the process, we are losing many of the valuable genes that are promising in the future breeding programs. Micropropagation, or a propagation method wherein we use a very small or micro-sized tissue from the parent plant, culture them under controlled conditions, and with the help of hormonal combinations, we try to multiply them. As seen in this image, you can see from a single tissue, we are able to generate hundreds of plantlets in the same vessel. And so this multiplication cycle goes on, and as much we can produce as many plants as we need. Of course, the standardization takes a bit of time and experimentation, but the better part of it is you can be able to restore it. The program is called eco-restoration. You can restore it back to nature from where these plants were lost. This is also a method that protects plants against all natural calamities, because these plants are grown under controlled climatic conditions. Additionally, when the tissue that we use is a meristem, it gives the added benefit of generating virus-free plantlets, which is a big issue in many of the crop that is used as food even. For example, tapioca, banana, and so on. Now, when we talk of loss of biodiversity, we have methods here where you can try to generate variability as well. So we have tried to do embryo rescue. When we try to hybridize between distantly related plants, there is all possibility that the embryo or the hybrid embryo gets aborted. But using tissue culture techniques, we are able to rescue these embryos by providing the artificial environment and the nutrients, and get the new hybrids as well. And when we culture the seeds, many of the plants are basically heterozygous. So when we grow the seedlings or generate the seedlings in vitro, the progenies express the heterozygous nature, and this image on the right bottom side shows the variability as done, or as we saw when we did the molecular profiles of the seedling progenies. So there was an ample range of diversity within the seedling progenies. So this method assures us a lot of diversity which can be generated in such controlled conditions. Because basically monoculture leads to disease vulnerability, plants fall prey to all sorts of pathogens, and this directly affects the agriculture production. Another method where we can generate diversity is protoplast fusion. As we know, living cells, especially plant cells, have a cell wall. But if we succeed to remove the cell wall, the advantage is we have a naked protoplast, which is amenable to all sorts of genetic engineering experiments. Whether you want to introduce a foreign DNA or you want to fuse it with another protoplast of a totally unrelated or a distant genera. So that is called somatic hybridization because we are doing the hybridization between somatic cells, not gametic cells as in nature. This is also being done successfully, and it is particularly used in our experiments. It was used to transfer or see whether desirable traits that are available in the wild or related species can be introgressed into the cultivated varieties. And we can see diffusing protoplasts here. Of course, it is again a lab-based experiments, and we can use fusogens to conduct these fusions or protoplast fusions. Now, when we generate so much of variability or diversity, it is our responsibility to standardize a method also for conservation of this diversity, and in vitro gene banks are the answer to this. They are the place where you can conserve endangered as well as threatened species and any important species safe in a small space away from the vagaries of nature. And whenever somebody needs it or the population is lost in nature, it can be brought back from the in vitro gene banks. So this is a practice, and we are trying to conserve whatever diversity we generated in vitro gene banks. At this point, I would also like to mention about the Svalbard seed vault, which is located in Norway. And the concept or the idea, the objective of the vault was to conserve the world's crop biodiversity safely against disasters. So it is already under ultra low temperatures in the Arctic region. And tomorrow, even if something disaster happens, the seeds are safe there for the use of posterity. This concept is also used to store pollen genome, which is actually haploid, but the male gamete, which is used in hybridization. So when we use or we want a male gamete containing the desirable traits, it may not be flowering at the same time as the counterpart or the mother plant. So this cryopreservation helps us to preserve pollen for a longer period, and later on for the desirable hybridization experiments, this can be hybridized with the cultivated varieties. The technology called artificial seeds or synthetic seed technology helps us to make encapsulated propagules, which are again disease-free and stored in controlled conditions, so that whenever a cultivator, a farmer, or a scientist needs it, because there is no more availability in nature, these seeds can be brought back to life and they develop into normal plantlets. Biofortification is also another need of the hour because it literally helps to breed health or bring in more health into the food that we normally consume. Conventionally, these breeding experiments would take years, at least 10 years, but with the advent of transgenic technologies, the gene editing CRISPR technologies, this has become faster. And this image on the top left shows a normal tomato that we all can consume. And you have the purple tomato, which is actually producing anthocyanins. So it is a biofortified purple tomato, which is expressing the genes that were inserted from the snapdragon plant. So this is a tomato with all its nutritional qualities, but with the additional biofortification of anthocyanin producing gene. We know that most of the crops are micronutrient deficient, which we call commonly as hidden hunger. So a lot of crops are being biofortified with the required micronutrients so as to help the betterment of health in general. Similarly, plants are reservoirs of medicinal compounds, we call them, and a lot of plants are still used for therapeutic uses. Molecular farming is one method wherein you can stop or at least reduce depending or destroying the plant population to a large extent. This is an example of the plant Artemisia, which is used to produce artemisinin, a powerful anti-malarial drug. So once we have standardized a method for their growth in vitro, We can use those plants, but if the metabolite is in the root, we can proliferate the root in vitro, or if it is in the whole entire body, then we can produce callus and make it go on producing or dividing more and more. And that helps us to produce the drug or the artemisinin slowly within a period of within a short period. The bioreactors also are the mechanisms wherein you can continue producing secondary metabolites, irrespective of depending or destroying the plant. Now, there are methods using machine learning or deep learning to predict diseases and also prevent them by using bio-friendly or eco-friendly organisms in the control or prevention of diseases. Some of my students who are working, one of them is working at the John Innes Center now, who's manipulating wheat grains so that they become red and they produce anthocyanins and tannins, which help them to become more resilient. And coming to the chemical fingerprinting, we are all familiar with turmeric or curcuma longa, but there are other related species. Example, like the black turmeric, which is very high in its medicinal properties. So we're trying to chemically fingerprint them and understand the mechanism of resilience and their adaptive capacities. Metabolite engineering is another important area where all the metabolites that help plants render their properties that we are exploiting can be engineered. The genes that lead to the production of these metabolites can be made to enhance their activity or even suppressed when they are not needed. So this helps to increase the plant immunity and make them develop stronger cell membranes and enhance the natural defense mechanisms. So we are actually alarmed to see the global hotspots of environmental AMR, and India is also affected by this AMR. So in short, I would like to say this conference was the need of the hour, and the most important part was the need for convergence of disciplines from people from different disciplines. And this conference has helped it to be one of this partnership. We cannot achieve anything without partnership. Thank you once again for this opportunity, for giving me a chance to remind that plant is a central pillar to the health of the globe. Thank you.
Thank you, madam.
Sorry, sir, I think the voice was
for your presenting your innovative ideas in correspondence with the Sustainable Development Goals. But due to certain sound problem, audience cannot be, some audience are complaining about that. We will record it and send to the audience later.
Oh.
Please remain, madam. And next we are moving to the last topic, but important. Fifth session. And fifth session is about the Kerala, how the One Health, we are facing the One Health problems. And with the motto of thinking globally and acting locally, we are conducting today's program. Let us have a glimpse at Kerala response to One Health problems. Kerala being an ecological sensitive area at the peninsular end of India, tucked between Western Ghats at east and Arabian Sea at west, Kerala is very sensitive to climate changes. Due to increased urbanization, population density, and population mobility, the probabilities of any new disease introduction as well as transmission is easy in Kerala. For example, first case of Nipah in South India was reported in 2018 in Kerala in our district. Now we are having yearly reporting Nipah, even though it was sporadically. And first COVID-19 in 2020 was reported in Kerala. It was imported from China. And first confirmed monkeypox case in 2022 from India was reported from Kerala itself, from Kollam district. Currently, amoebic meningoencephalitis are reporting from most of the districts from this state. Same time, in India, One Health state program was first launched and materialized in Kerala. So next, we have to know what are the One Health threats, what are the preparedness of Kerala. So for this presentation, I invite Dr. Anish, Nodal Officer, Kerala One Health Center. Over to Anish.
Yeah, thank you, sir. Thank you for inviting me and for the nice introduction. I will share my slides.
Please wait.
Sir, do you want to close the slides?
No, you can continue. Please change it to presentation mode.
Okay. Okay. Am I audible, sir?
Yes, you are audible and visible. Continue.
Yeah, thank you. So I am speaking specifically about the Kerala's context and Kerala aspect of One Health threats and preparedness. And I will just go to the slides. So this slide is partly explained by Sir, because we are very special in the One Health platform or One Health context of the globe itself. So one is we have got very specific geospatial concerns, as Sir has mentioned. This one side it is Western Ghat, and the other side it is sea, and it is a very narrow spectrum of land with almost 300 centimeters of rainfall. And it is just like a tropical rainforest, but it is grossly populated. So the climate and climate change, if you just look at the impact of climate change also, you can see that Kerala is facing a lot of challenges because of the climate change. So population density and population distribution, Kerala is maybe fourth in the population density in the whole country. Maybe 1 decade before, it was the first, but now it is the fourth. But still, if you just look at the population distribution, you can see that the entire Kerala, the population is distributed entire Kerala, making it maybe a largest city in India. Kerala as such, maybe as a whole, it's the largest city in India. So migration and population movements is also a big challenge. And Sir has already mentioned that how we detected COVID-19 and how this Mpox was detected. Out of almost 40 cases detected in India, more than 50% of Mpox cases are from Kerala. So that is a situation that we are facing related to the population movements. Population movements, not only the diseases are coming from outside India. If you just look at the maybe in-house population movements or maybe domestic population movements, you can see that so many people from different parts of India is coming to Kerala because of job and all. And these states are facing maybe the old world infectious diseases. And Kerala is prone for those kinds of diseases, including malaria and filariasis. And if you look at the vulnerable population, if you just look at individual vulnerability, you can see that Kerala is the diabetic capital of India. Kerala is having maybe the elderly proportion is double that of an Indian average. And still, if you just look at the social vulnerabilities, then you can see that we have got a significant proportion of people, tribals, maybe people residing in coastal areas, and maybe these dust workers and all other things. And rapid urbanization is making a big problem, and we are building maybe towns, maybe inside forest, and there may be fragmented ecosystems, and human wildlife conflict is also a big issue. If you just look at the emerging diseases, you can see that a lot of diseases are still in Kerala. Almost all districts in Kerala has reported the high priority avian influenza in birds. And if you look at the Nipah, you can see that it is affecting maybe 5 or 6 districts in Kerala. And even though it has started from Calicut, it is maybe moving towards the south, affecting Malappuram and Palakkad districts now. So if you just look at another BSL-4 pathogen, that is KFD, you can see a couple of districts in Kerala, that is Wayanad and Malappuram, is affected by KFD. And so many other diseases are emerging, just like Sir has mentioned about the amoebic meningitis. And this is the situation of emerging infectious diseases. On the other hand, if you look at the surveillance and health system attributes, Kerala is maybe having the most important or the best surveillance mechanism in India, so that we are able to capture these diseases. And that is actually something beautiful, that Kerala can capture diseases and it can prevent spillovers, so that we may able to help people, or maybe able to build resilience against maybe large outbreaks of infectious diseases. This is a picture that you are seeing in the left upper side is Brahmapuram fire incident. And what you are seeing in the lower side is our minister of LSGD is batting in the pitch, which is actually made at the Brahmapuram itself. The picture that you are seeing in the upper maybe left corner is that of 2 years before, and what the picture you are seeing in the right down is picture now. So I am actually putting this Brahmapuram also. When we are thinking about One Health is not pertaining to wild health or wildlife human interactions only. It is, as we have already discussed about the plant biology, as we have discussed about the prebiotic, postbiotics, and all. These kinds of things are also important. So the climate change will bring these kinds of issues, that the legacies, urban waste can catch fire, and a lot of organic substances like maybe persistent organic pollutants can go to the earth, and it can get into human food chain, and it can create a lot of issues. So in Kerala, it is not only the Western Ghat, the coastal area is also important, because these kinds of waste, maybe inorganic pollutants like mercury and all other things, will go to the sea, and it can get incorporated to fishes, and it can go back to human. So whenever we are thinking about One Health, we have to incorporate all these aspects also into the One Health. We cannot limit it to maybe to the wildlife spillovers and infectious diseases alone. So if you just look at the One Health initiatives of Kerala, I am just making a small list because of time constraint. It starts from the Center of One Health Education, Advocacy, Education, and Training, that is cohort, that initiated by Kerala Veterinary University and Veterinary and Animal Science University in 2013. So whenever you are just looking at the initiatives of One Health, you can see that all the initiatives started from the animal side first, and then it was incorporated by the human side. So the Nipah surveillance and control activities of 2018 is a yardmark, or maybe a milestone in the One Health initiative of Kerala, even if we were not calling it as an One Health surveillance system. But Nipah in Kerala is always an One Health system, because Nipah is a typical One Health disease where the wildlife, maybe domestic animals, human health systems, environment, and climate change, and all other things are just taking or making a role. So maybe Nipah is the context where we were telling, or we were discussing about One Health. Dr. Arun has already mentioned. And a community education program of One Health was started by Kerala Shastra Sahitya Parishad in 2020, just maybe soon after the peak of COVID-19 outbreak, because the state subject committee of Kerala Shastra Sahitya Parishad was seeing that this is a big concern, maybe an emerging concern. And Dr. Arun himself drafted the first slide. We were using 3 slides, and we were conducting extensive educational campaign across Kerala. And Dr. Jayakrishnan sir is also involved in these activities. And after that, One Health program was initiated by Government of Kerala, piggybacking to the Rebuild Kerala initiative in 2021. You may be knowing that Rebuild Kerala initiative is a government program funded by World Bank soon after the flood of 2018. The program started in 2019 itself, but health was not included in the Rebuild Kerala initiative in the first phase. But soon the COVID-19 has come, and so we recognize that health is not only a welfare sector, it is a developmental sector also. And we identified that there should be One Health program, and better, it may be One Health program. So that is why Kerala is the first in the country, and one of the first few communities in the entire world initiated a One Health program, incorporated to a government program or a community-based program. So it was as a part of Rebuild Kerala initiative. And it started in 4 districts in Kerala. That is a Pamba Basin district. That is Kottayam, Idukki, Pathanamthitta, and Alappuzha. And still the program is running. And I will just say some words about the Rebuild Kerala initiative, part of One Health program. And in 2023, Kerala Public Health Act, I am stating that for Kerala Public Health Act, if you just read the preamble of Kerala Public Health Act, it recognizes One Health as a very important mitigation strategy against the outbreaks and infectious diseases. That is one aspect of that. Second one is Kerala Public Health Act. The authority, the Public Health Authority, was shifted from an officer to a body, that is a democratic body chaired by an elected leader. The local public health authority is chaired by the Panchayat President. The district this public health authority is chaired by the district Panchayat President, and the state public health authority is chaired by the state health minister. So this public health authority is composed of people from all one sectors. Maybe apart from human health, there are people coming from animal health, maybe agriculture, water, and environment, forestry, and so many, or food security, food safety. And all these people are incorporated to the Public Health Authority of Kerala by the Act 2023. So now Kerala is moving to escalating the One Health activity to the entire state. Now also it is there in a piecemeal fashion, but as a program, it is known as Kerala Health Improvement Program. It was initiated already in 2025, but it will be completely running by 2026 onwards. By that time, the entire Kerala will be covered by the One Health initiatives or the government-initiated One Health program. So some dedicated activities in One Health program. One is One Health volunteer network. So Kerala is trying to capitalize the volunteerism that is present in the community, so that everybody can act as a One Health volunteer. They can report maybe unusual symptoms or disease spreading, or maybe even, for example, in KFD, if you are just witnessing a monkey fall, even you can report that. And community-based surveillance and joint investigation strategies is another component. And for example, some diseases were identified as One Health priorities, just like Nipah, KFD, maybe avian influenza, dengue, even though there is no animal component still visible, but still it is present, rabies, and all other things. We have identified maybe a dozen of One Health priorities. And for all these diseases, there is a guideline for surveillance and joint investigation strategies that is already prepared, and it will be as a part of this Kerala Health Improvement Program. This will be running shortly. And another is strengthening of surveillance mechanism through hub and spoke model. All the laboratories, especially the government laboratories in Kerala, is networked under Nirnaya, that is a dedicated program. And we are discussing with the private parties to incorporate them also into this particular model. Even the laboratories that is present with animal husbandry, the laboratories again present with maybe water authority, and you may be knowing that Kerala is in Ernakulam. We have got the first laboratory by Pollution Control Board, which will evaluate the AMR, that is antimicrobial resistant pathogens in the wastewater. So that kind of laboratories will be incorporated. So that kind of a lab network system will also be there. And we try to institutionalize the One Health activities. It is governed by the nodal agency, that is Center of One Health Kerala, that is situated in Trivandrum. And there is Kerala Antimicrobial Resistance Strategic Action Plan. It is a dedicated activity, that is CARSAP, for antimicrobial resistance. By just recognizing that antimicrobial resistance will be a large future challenge. Maybe it may be going to be dominating all other One Health challenges in future. And another is, as we are facing a special risk of Nipah. We have got Kerala One Health Center for Nipah Research and Resilience situated in Calicut. Still, now I am delivering this presentation from the center. And we have got Kerala Center for Disease Control and Prevention, that is KCDC, in the model of the US KCDC, we also developed a Center for Disease Control and Prevention in Kerala. And it is a couple of programs were started, but it is not in full swing. And again, we have identified or we have institutionalized so many other institutions that can support One Health initiatives, even if it is not maybe it is not under health department. One is the Institution of Advanced Virology, IAV, and Thonakal, Thiruvananthapuram. Second one is Kerala State Climate Change Adaptation Mission, Center for One Health Education that I have already mentioned, that cohort. And there is a program known as Vitut. Vitut is the seed shower. The seed will be made to bowls and it will be put maybe in fringe forests for us to attract maybe bats and other animals, so that the bats and other animals may not be interacting to the human dwelling places. And One Health and LSTD initiation program. There is some implementation researches are going with KILA, that is Kerala Institute of Local Administration, and so many other activities are in CWDRM is there in Calicut. They are also doing a lot of works in water-related diseases, and so many things are happening. And I am just giving a list of something. And among these initiatives, one thing is very special, has got special mention, is Kerala Antimicrobial Resistance Surveillance Network. That is CARSNet. That is a part of CARSAT. And you can see that is the report of 2024. And in right-hand side, you can see different hospitals contributing to this particular antibiogram. So you can see the entire Kerala is almost covered, and every day new and new hospitals and labs are incorporated to the program. And we are continuously evaluating the priority pathogens for the antimicrobial resistance. And we are seeing that the antimicrobial resistance is more in tertiary care hospitals, where the antibiotics are maybe the reserve type of antibiotics are used in high proportions. And PHCs, the antimicrobial resistance is low. And we can just map where the disease is or where the antimicrobial pathogens are transmitting, and how much, what is the gravity of these things, how it is reversed, and all other things. So these kinds of networks and governance and surveillance system is already present. And you may be knowing that there is an Operation Blue, that is the blue is the color of AMR, and you are not able to purchase the antibiotics without a prescription in Kerala. Sometimes you may be seeing some pharmacists may not be following the rules, but definitely almost 1,000 crores of rupees were saved by preventing the prescription of unwanted antibiotics last year. So that kind of activities are happening in antimicrobial resistant picture also. And there are other new initiatives are there. One is EpiForm. EpiForm is the epidemiological platform for prediction and forecasting of infectious diseases. And with the help of the National IT, Ministry of IT, we are developing that program. Primarily now it is for 4 diseases, that is dengue, leptospirosis, Nipah, and influenza. But maybe we may be extending this program to other diseases also, where we are trying to have some disease prediction models, both the automated, maybe the statistical models and compartment models and we are trying to incorporate AI also to predict diseases, so that we can be more prepared for the outbreaks. Second one is a public health intelligence system. It is a part of this KCDC. We are trying to evaluate what is happening all over the world and what is the proportion of Malayalis there, what is the possibility of a person harboring a disease and coming back to Kerala, and all other things are continuously evaluated. That is public health intelligence system. And in 2 occasions or in 2 areas, we are maybe in a pilot project, we are trying to initiate a wastewater surveillance system. One is in Trivandrum and second one is in Calicut. Trivandrum, it may be for hepatitis A, because Trivandrum is the only part in Kerala where we have got maybe a sewage treatment system, maybe a community-based sewage treatment plants and systems. So that is why we are isolating, or we are dedicating this particular project to Trivandrum. But in Calicut, there is an issue that you may be knowing that Nipah is a concern. And sometimes a Nipah case may be reported in some hospitals in Calicut. And if it is undiagnosed, we can detect maybe by a hospital-based surveillance, wastewater surveillance system. So those kinds of thoughts are happening, and these kinds of systems are about to be built. And as I am representing the Nipah One Health Center, I want to say something about the Nipah specific investigations that is happening. And whenever there is an outbreak of Nipah, what we are challenged is the community's response, the inadequate community response, especially from doctors. Some of the doctors will say that, okay, we know that Nipah is a self-limiting outbreak. It may just go to maybe 10 or 20 people may be affected. It's a maximum. Most often it will be end up in a single case. They're saying what they are saying is right in partially, because that is the picture of Nipah so far. But you have to keep it in your mind that the whole history of Nipah, we have got maybe 30 years of history. Even if you are just searching from the Hendra infection of Australia itself. So Nipah is a fastly evolving virus, and it is an RNA virus. It is the Henipavirus family, and it is very, very prone for mutations. And every spillover cases is an opportunity for Nipah virus to mutate itself, and it can be turned to a human virus. So Kerala, we have got maybe some responsibilities for the entire world, that we have to check the Nipah virus turn to be a pandemic virus, because Nipah is one of the 10 diseases identified by WHO, which has got huge pandemic potential. So that is the importance of Nipah. That means each and every Nipah case is important, and it is very important that you prevent each and every spillover, each and every spread. So that is why we are trying to find out every people who has got a potential of infection and try to isolate them. And even we are trying to regionally maybe restrict maybe for a few days and all. It is based on some suggestions or maybe advices from the national bodies also. Anyway, so that is the case of Nipah outbreak. But we are trying to conduct a lot of investigations in Nipah. Dr. Arun is also part of in some of them. One is a study on transmission mechanisms, because in Kerala, what we are lacking behind is we do not know exactly what is the transmission mechanism of Nipah. So with the help of National Institute of Virology and National Institute of One Health, we are trying to study the transmission mechanisms of Nipah. Every month, Dr. Arun and his team is coming to Calicut and other adjacent districts to see or to get samples from maybe different sources. And another is a pseudovirus study among animals with the help of IAV. Pseudovirus means it is actually the virus which will mimic. It is not an infecting virus, but it is a viral structure which can mimic maybe the Nipah virus. If the antibodies against Nipah is present in your body or the body of animals, as it is neutralizing Nipah virus, it can neutralize pseudovirus also. So using pseudovirus, we are checking whether some animals harboring the antibodies against Nipah virus. So that kind of an activity is happening in Palakkad now. And we are trying to study the bat behavior by using drones with the help of ICMR headquarters, National Institute of Epidemiology, IIT Madras, and Kerala Forest Research Institute. And climatological study is carrying out with the help of Kerala State Disaster Management Authority. And now we are on the verge of an implementation research with the help of Kerala Institute of Local Administration to prevent Nipah spillovers or to build resilience against Nipah. And sometimes you might have known that Nipah vaccine is under trial. The first phase of trial is already commissioned, and now they are moving for the second phase of the trial, where exactly you may be knowing our Covishield, that is the AstraZeneca, this Oxford University vaccine, which is using vector vaccine. The chimpanzee adenovirus is used as a vector to send maybe the spike protein of COVID-19 to your body. Similarly, we are using some of the antigens that is present in Nipah virus, using the same vector, that is the chimpanzee adenovirus. So that vaccine is almost ready. It is entering to phase 2 trials. Maybe Serum Institute of India maybe the vaccine distributor, that vaccine manufacturer and distributor. And this is the time that we can do some vaccine studies, the vaccine acceptability studies. For example, if there is a vaccine is available, how much our community or people will be willing to accept those vaccines. those kinds of studies can be done. This is a very good time for that. Even KMCT, with the help of maybe we can collaborate with KMCT to do these kinds of studies. So these are the activities that is going on in NIPA nowadays. And these are the things I wanted to discuss, and definitely I will be happy to take questions and discuss further. Thank you.
Thank you, Anish, for your enlightening presentation, especially about Kerala, the hotspot of One Health. And today, in the international webinar, people from many countries are attending. And first we start with India and last end with Kerala. In between, we talk about zoonotic spills, and in between, the third and fourth presentations are very innovations, how to tide over the One Health problem. One is about the gut flora, second is from the plant field. On behalf of KMCT, I once again thank to all the presenters. Now we can go for a queries. Please, the panelists, please be ready. We will read the questions and you have to answer that briefly.
So with this, we have come to the end of our scientific session, and we have received a few queries from the audience. I request our speakers to address these queries. I shall share the queries here. First to Dr. Arun T. Ramesh. I hope Arun sir is still in the call.
Yes, I'm
Sir,
here.
yes, sir. First question is from Dr. Vijesh Venugopal. What would be the top 3 One Health interventions that you would like to implement presently?
So the priority will be one intersectoral surveillance, like that I've mentioned in my study. Under the National One Health Mission, we are already studying different kind of animal-human interfaces. There are livestock human interfaces and wildlife human interfaces at zoological parks, and in forest range areas we have a very different kind of interface. So it's very important to detect a spillover at the interface itself. So that is one of the One Health priority. And other thing is joint outbreak investigation. As Anish sir has mentioned about Kerala, all over India, there is a NGORT, National Joint Outbreak Research team that has been established. So that's another priority. And third will be antimicrobial resistance. That will, the third priority will be the surveillance of antimicrobial resistance.
Sir, thank you. Sir, next question is from Dr. Dinesh Kumar. One Health means all living organism on one side and human beings on another side, or healthy environment for all living organism.
Yeah. So like One Health means recognizing the interconnectedness of human health, animal health, and environmental health. So it's like we are considering the entire environment as a single unit. I can admit that still our One Health initiatives are bit anthropocentric. We are more concentrated upon the zoonotic diseases or like the zoonotic threats that humans are facing. Yeah, it's still anthropocentric, but like One Health is considering everything like it's like about a planetary health, or you can say we are considering the entire environment as a single unit.
Okay, thank you so much, sir. Next, we move on to queries to Dr. Mohammed. First question is from Dr. Sunanda. How does climate change accelerate zoonotic disease spillover?
Yes. As we discussed in the presentation, the zoonotic spillover is primarily based on the mutations, primarily causing one, increased virulence for the pathogen, or reducing the resistance, the tolerance or resilience of host species, including humans. Climate change or environmental change is a major trigger in causing mutations, these mutations. So, for example, a single mutation, which we call it as single nucleotide polymorphism, which simply it means that there are 4 nucleotides in a DNA: adenine, thymine, guanine, and cytosine. If a single adenine replaced by thymine, that is called a single nucleotide polymorphism or SNP. Even a single SNP can cause increase in virulence in a pathogenic organism. So that will change into a highly virulent organism and cause a pandemic or a spillover in some cases. We have conducted extensive studies on these mutations. Interestingly, we found that some of the mutations historically that took place over thousands of years. Recently, due to climate change, these mutations are taking place within years, within decades. So that is really an alarming situation. Especially, we studied specific traits in animals. Say, for example, we studied body size. Historically, over millennia, on an evolutionary basis, we can see that body size is increasing. So, increasing body size is a trigger for mutations. What does it mean? It simply means that if a species increases in its body size, naturally they are becoming more and more prone to these infections and become less resistant and less tolerant. And yes, this is the basic reason why environmental change is causing increased zoonotic spillover and pandemics. There is an interesting article we published back in 2018. You can just read that. And also, which is available in my Google Scholar profile. Size does matter. We proved it with our small Vechur cattle, with our large Holstein Friesian cattle. Yes. And the rest of those things, you can just follow those articles. Great.
The next question is from Dr. Ashwati Vinod. How can we ensure community level awareness of zoonotic disease prevention and environmental health?
Yes, that's a good question. The basic thing in increasing the awareness is understanding the basic mechanisms, what is happening in between, how SARS-CoV-2 virus happily living in bats or wild animals, all on a sudden, how they gain entry into humans and how did they become pandemic. So increasing awareness, that too at the basic level of scientific evidence, that is the best way for increasing community awareness. Yes.
Thank you, sir. Next, we have 2 questions from Dr. Danya Muralidharan. First one is, why is saliva of animals a source of zoonotic spillover? And also, she has asked, what is the role of amplifiers in this triad?
Yes. In case of a source of infection, saliva in most of the cases, it is not only saliva, but also blood, milk, or any body secretion is a source of infection. But for most of the neurotropic viruses, they get entry into the saliva so easily, and saliva is a good medium for their growth, multiplication, and development. So whenever you are getting access to the saliva, thus it becomes a most common source of infection. Definitely, if you have access to other body fluids such as blood, which is very less common in most of the cases, that is why blood or other body fluids are less common in spreading the infection. Yes, we should be very careful when we get in contact with the saliva. Most of those pathogens, they transmit through saliva. What is the role of amplifiers in this triad? A good question. I understand you were following the presentations in this session, because basically the zoonotic spill or zoonosis happen when these organisms become virulent. In that process, in some of those cases, most of the wild animals are reservoirs, so they do not amplify or they do not enhance the virulence of the organism to become it more pathogenic and infect humans or other animals. So they control it literally. But when they gain entry into a domestic animal, so they are free. Domestic animals, they are not very familiar with these pathogens, so they cannot immediately control these multiplication or their development. Hence, these organisms amplify in the specific host. So we have identified certain, I have already explained in my presentation, that there are specific amplifiers like pigs or poultry in case of avian influenza, these specific amplifiers, and we currently connect these amplifiers with the transmission dynamics of specific diseases.
So we have one more query from Dr. Asman Faiz. Do emerging zoonotic diseases exhibit early or unique oral manifestations that dentists should be trained to identify?
Yes, exactly. As Dr. Thanya rightly mentioned, and I explained, saliva is a major source of most of the pathogens. So our dentists and dental surgeons should be trained to deal with this particular body fluid because, say, for example, classically for rabies, rabies directly enter into the saliva, and that is the highly infectious form of virus in most of the cases. So even if initial stages of non-symptomatic form of disease, these organisms may be there in the saliva. And for other emerging pathogens like Nipah or even for COVID-19 or Ebola or some other pathogenic organisms, we are still in the process of identifying how the salivary excretion is and how the saliva plays a role in transmitting these diseases. So definitely it is a concern and an area of further investigation, and we should identify the possible threats and take ameliorative measures. Yes.
Thank you, sir. Next, we move on to the next query. What happened? Next query is to Dr. Deep Jyoti Boyan. So first one is from Mr. Ajmal M. What is the role of postbiotics in management of halitosis?
That's a very good question as well. Yeah, halitosis is essentially bad breath. So, I mean, I think Dr. Mohammed also outlined the importance of saliva in zoonotic diseases. The same goes for, gut microbiome related diseases. So oral health is quite important. So is the oral microbiome. So what happens is that if there is any sort of dysregulation in the gut microbiome, it can actually affect your oral microbiome and vice versa. And that can lead to halitosis. Yes, there has been some studies, but most of the studies that have been done are on probiotics. So basically the live bacteria. But there was a study, I think, back in 2023, where they looked at both the probiotic and postbiotic versions of a bacteria called Lactobacillus paracasi, and they found that both the live and the dead bacteria. So when I say dead bacteria are also considered postbiotics because they don't have the live component to it. So yeah, they found that both the live and the dead bacteria were equally effective in controlling halitosis and also changing the oral microbiome to a healthier state. So they saw a regulation of or reduction of organisms like Streptococcus in the oral cavity, which basically means that it was able to reduce the halitosis as well as improve the oral microbiome.
Thank you, sir. Next question is from Dr. Rita James. Unlike prebiotics and probiotics, where postbiotics are generated in vivo, will postbiotics supplied from outside reach the desired site of action unchanged or in the desired form?
Again, very good question, Dr. Rita James, and thank you for that. Yes, postbiotics are produced in vivo, basically within our gut, and most of the times they do quite a lot of work in terms of the physiological functions of the body, maintaining gut integrity, reducing inflammation, modulating the immune system. So yes. There are different ways to look at this, basically how you could improve postbiotics in the gut. So of course you can do external formula where you can use different encapsulation technique and different nanoparticle as well as liposomal based delivery systems, where you can directly deliver these postbiotics to the colon or your site of interest. So in our case, we are also experimenting on how we could use different delivery system to directly deliver postbiotics in the tumor microenvironment for gastric cancer, breast cancer, and lymphoma. So that actually helps. So different formulation techniques can be used to supply postbiotics to the desired site of action.
So next question is from Dr. Navya George. Is there any commercially available postbiotics?
Yes, there are quite a few commercially available postbiotics. In Australia, things are a bit different because Australian Therapeutic Good Administration is stricter than the FDA in the US. So in the US, you can find short-chain fatty acid formulations, which are considered postbiotics, of course. And also now, as we also consider dead probiotic bacteria as postbiotics, so there are a lot of commercial products coming up, and we have been working with quite a lot of commercial manufacturers as well to bring those new postbiotics, essentially the dead probiotic bacteria, to the market. I'm not sure about India, but I think the best way to have postbiotics, as I said in my talk, would be to eat more fermented foods. Because you're getting both all three things together, so pre, pro, and post, and that's probably I would suggest having that option rather than going for supplements, because supplements are not very well studied and might not work as intended. Food is probably the safest and the easiest way to improve postbiotics in the gut, as well as to improve your gut microbial communities.
Oh. Next query is from Dr. Sunanda. What all could be the potential side effects of postbiotics?
Really good question, and that is something that field is now studying. So we are currently working on a number of postbiotics that are considered bad postbiotics, like the one that I showed you earlier in my slides, where if you consume a certain Western diet that is high in fat and carbohydrates, it can lead to production of postbiotics like TMAO, which is a toxic metabolite, or can be labeled as bad postbiotics, and that has a lot of implications in heart diseases, diabetes, and atherosclerosis. And there are things like bile acids as well. Secondary bile acids are also produced by the gut microbiota, and they can be considered as toxic metabolites. So you have to watch what you eat.
So there is one more new query in the chat box. It goes like this. Can microbiome modulation be integrated into personalized medicine for preventing age-related diseases? And what are the challenges in moving microbiome-based therapeutics into clinical practice? Could Western diets influence telomere dynamics differently compared to traditionally Assamese rich in fermented foods?
Oh, wow, that's three questions, very long questions. I think to answer your question, yes, gut microbiome based therapeutics can be integrated to personalized medicine because, as we know, just like our DNA, our gut microbiome signature is unique. And as I previously mentioned, about 57% of our body cells are bacterial cells. So you need to cater that when you are looking at personalized medicine, especially in terms of cancer and things like that. What was the second part of the question?
Second part was like, what are the challenges in moving microbiome-based therapeutics into clinical practice?
There are quite a few challenges because, as you know, I mean, gut microbiome-based therapies are there for a long time. It's like when it comes to especially prebiotics and probiotics. And now we know that there has been enough evidence on those two components. So pre and pro. So if you eat fiber-rich foods and if you eat probiotic foods, there are, you are going to modulate the microbiome. But whether or not that modulation is to a clinical significance, that's something that we don't know yet. So we need more research. But when it comes to postbiotics, as I said, it is an emerging concept and more research is happening in that space. Especially, I think a lot of research studies have shown that the impact is predominantly, so impact is measured predominantly using preclinical models. So there hasn't been a lot of clinical studies. So we do need those. We, of course, need the preclinical evidence to inform the clinical studies. But yeah, in terms of some postbiotics that have been currently investigated for infectious diseases like Clostridium difficile and IBD and IBS as well, there has been quite a lot of studies on that. But yes, clinical studies, longitudinal studies, randomized controlled trials are probably needed before we move on to especially postbiotics. But prebiotics and probiotics are already proven in many instances that they actually can do a lot of health benefits. And the last one was about, I think, fermented foods of Northeast India.
Yeah, it was like, could Western diets influence telomere dynamics differently compared to traditional Assamese food?
Yeah, I mean, it's quite interesting that you mentioned Assamese. So yes, so telomere length is linked with age-related disorders. So we know that, as we age, our telomere length shrinks. And yes, Western diet rich in fat and lack of fiber, for example, so they can impact the aging process negatively, and they can actually reduce the length of the telomere. Fermented foods, on the other hand, can have both direct and indirect effect. So direct effect being, I won't be able to tell you on Assamese diet specifically because there hasn't been a lot of studies looking at telomere length and Assamese fermented foods. But in general, fermented foods are considered healthier options because they are more bioavailable. So every nutrient that we get in fermented foods, be that antioxidants or phenolic compounds. I think Dr. Meenu talked about phenolic compounds and importance of phenolic compounds in health overall. So in fermented foods, you get those, you are able to absorb those nutrients better because they are already broken down by the probiotic microorganisms that are present in the fermented food, which basically leads to better bioavailability and which essentially leads to better bioactivity or preventing a lot of diseases as well as helping in treatment of different diseases. So I won't be able to tell you exactly about the telomere length in this context, but I can tell you that fermented foods, if prepared correctly, so you have to be careful how you prepare fermented foods because there are a lot of risk of contamination and there are a lot of cases around the world when it comes to people dying from fermented foods because they were contaminated, right? So that quality control aspect is quite important. And traditionally prepared fermented foods like dosas and idli that we already know are safe. So that can be consumed. But when you are experimenting with new fermented foods that are not from the country, let's say you want to try sauerkraut or you want to try kimchi from Korea. And first of all, I think there should be a standardized process to make the fermented food so that you can reduce the risk of contamination. Yes, if you are preparing your fermented foods properly and they're not, they don't have any sorts of contamination for external pathogens, they are good for you overall compared to unfermented counterparts or compared to Western foods like burgers and pizzas.
Thank you so much, sir. Now we move on to next
Thanks.
query. Yes. Next query was to Dr. Minu Divakaran. It was already answered in the chat box. The question was like, ma'am, can these pollen be used positively to create antibodies like we use attenuated viruses so that it can be used as a vaccine in hypersensitive individuals or in allergy treatment? Ma'am, would you like to add anything to it?
No, I think I answered in the chat box because it is being done already. That was a very interesting question. And since it is in the immune responses, I didn't give more details on that, but it is being done in crops like lotus, the dandelions, and the date palms even. Primarily because pollen of any crop has a very strong exine. That's why it's also used in fossil studies, evolution studies. So the exine can resist even acids. So that is the reason it is being used in allergy studies also. I think the possibility, I'm clear about the possibility, Dr. Harish.
Yes, ma'am. Yes, ma'am. Can we move on to the next query? Thank you so much, ma'am.
Thank you.
Next query was to Dr. Anish from Dr. Asman Faiz. How can dental research help us understand how pathogens spread through saliva and aerosols during pandemics in the future?
Yeah, thank you for that question. I think it is partially addressed by Dr. Muhammad. It depends on how the pandemic is spreading. Usually, a pandemic, especially a viral pandemic, will be airborne. An airborne or an aerosol-based disease transmission, in those cases, saliva may be very important. So those kinds of, as we are understanding from the COVID-19, as we have got some concept regarding how the body fluids are affected during pandemics, maybe including not only the saliva, maybe lacrimal secretions and all other things. Definitely, these things could be used as a early warning system, or maybe dental doctors when they are seeing patients, and if they are collect some samples for saliva, it could be studied for the further mutations during pandemic spreads and all are very important. As this may be one of the lesson that we have learned from COVID-19 pandemic. That could be definitely incorporated into surveillance mechanisms during pandemics, especially if it is transmitted through airborne or aerosol based. Thank you.
Thank you so much, sir. And with this, we come to the end of the scientific session and the webinar. The link for the e-certificate has been shared in the chat box. Please fill out the Google form using the same, and your participation certificate will be sent to the email ID you provide in the form. Once again, our sincere thanks to everyone for being part of the conference and making it a meaningful and a memorable event. And on part from the organizing committee, I extend heartfelt gratitude to all our distinguished speakers for sharing their expertise, insights, and experiences with such clarity and depth. Once again, thank you all and have a very good evening. Thank you all.
Thank you.
Thank you. Thank you.
Thank you.
A QR code is displayed on the screen. You can also use this QR code for the link for the e-certificate for receiving the e-certificate. You can also scan the QR code or use the link that is being given in the chat box.
you.